It's Graeme though, isn't it? Yeah, right. Okay, fine. I think we're all squared away. We already got the presentation up behind me, so that's good. The first of our series now since coffee break, and we've got another 6 that we're going to go through. It's from CSIRO again, Scott Chapman and Graeme Hammer. Graeme's going to present this paper. It's entitled Extending the Phenotype, Integration of Field and Glasshouse Phenotyping with Crop Modeling. Graeme.
Okay, thanks very much. I'm sure you can all tell fairly quickly that I'm not Scott Chapman, and he apologises for not being here, but he did give me 500 slides and say, off you go. So, well, for those of you who know Scott, that's not far from the truth. So, what I want to cover in this sort of extending the phenotype with modelling bit is just a bit of background on genetic improvement, and I think you've already had most of what I'm going to say, I think, but just look at what's plant breeding about, a little bit about complexity, physiology and models, and then a bit more on the roles of models in field phenotyping and trying to make the connections, and I think Francois did a really nice job of explaining all that yesterday. So the genetic improvement piece, it's very good for an agronomist, physiologist to get up here and say what the breeders do, but this is sort of my perception of what they do, but I think the KWS talk sort of helped with that. So if you look on the right side here, you've sort of got this, pipeline. So you've got some sort of parental selection and crossing, some novel germplasm coming in, some selection in these progeny, multi-environment testing as you screen those progeny and go through that pipeline, ultimately leading to some release of cultivars and back around to best lines maybe coming back into the parental pool. A whole lot of prediction and phenotyping going on in here as you go through this, because really prediction is what you need to understand which of these genotypes to advance, and the schematic on the left really just says the same thing. So the plant breeders can kill me later. And the other important thing to have in here is this concept of genetic gain. And so this is really what the whole breeding game is about. So this rate of genetic gain, R, is really deconstructed by these things. The selection intensity, so what percentage do you keep? The heritability, how much of the variance is genetic, the phenotypic variance for the trait you're looking at, and the number of years you can fit in, the number of cycles you can fit into a year. So this is genetic gain per year. And critical in that is this concept of prediction. And I don't know if Fred Van Nieuwek's still here, he is. But Fred, this was 20 minutes yesterday, so I got out of Fred's talk this equation. Phenotype for genotype I and environment J is something that looks like that. And you can then complicate it with all different perturbations. But for the challenge for physiologists is really going from, and Greg Rubeski said it the other day, if it's about yield, just measure yield. But there are lots of component traits that contribute to yield, but you're looking at correlated responses or component traits rather than the actual direct response. So you've got to keep this background in mind while you're thinking about what can we do with phenotyping that adds value to this, and can modelling do anything. And that brings me to the next point on complexity, physiology and models. And this is another one of Scott's pictures where the issue is really about there's multiple scales of biological organisation that we're working across. So if you start in the nucleus and the genes here, and you do some sort of, you get out to the cell with expression of those genes, and ultimately out to organs and plant with some expression of traits that get you to crop growth and development. And this is sort of a massive scale difference from molecular right through to crop. And there's a lot of context dependencies in here. So genotypes don't act in isolation, and different environments have different effects. So you get all these context dependencies, and the it depends remains a very pertinent phrase. So these context dependencies constrain molecular breeding, they constrain gene-to-phenotype prediction, and using either statistical methods or genomic prediction models, because they don't deal with it all that well. So this is where crop models can help a little bit, because they can start to help you think about how to span that phenotypic distance, particularly for complex traits. So I sort of like this picture, where I try to think about where does a model fit, because this picture puts it in the middle, and that suits me. But it's more about thinking of getting from this prediction from genetic space to phenotypic space. And a model can really help you in a number of ways. It can help you by predicting what consequences of trait variation might be, and you've seen some of that, and you can do that in target environments. And you can also then use the dynamic crop model framework, which does crop growth and development over time, to help you dissect and understand simple traits, because it points to the things that underpin them. You can then use that to detect regions responsible for those, and that may be where phenotyping comes in. And Vincent's talk was great about, I've got a target and this is how I need to go to phenotype for it, because it depends on canopy development and canopy conductance. Once you understand that genetic architecture, you can bring it back into the crop model and do all these things. So it's it's just a framework that does nonlinear interactions of crop physiology, crop growth and development, that can help you overcome a lot of these context dependencies. So if we look at a couple of examples in field phenotyping, I wanted to cover canopy dynamics, and this is the first point here, looking at genetic parameters for leaf area, and this notion of combining glasshouse models with in-field phenotyping. And so there were some questions about that earlier. I think it's very critical that these things can really happen. There are things you can do in glasshouses that much better you can do in glasshouses than in the field, and I think Francois showed that beautifully the other day. And you can do reverse modelling from field-derived data to get some of these things. So that one, you've seen all the pictures from Bernie the other day about all the phenotyping tools for the field platforms, so you know all that stuff. Scott's been playing around with this aerial imaging platform in Queensland and has developed a very nice, again, I think Melba made this point. You've really got to have your data curation, processing really sorted for these things, because you generate, I can't use the Australian terminology, but what shall we say, a lot of data, and you've got to get it right, and you want to get it back to plot scale things that you can use to do things with, rather than just an index at a point in time, which I think was also KWS guy's point and Daniela's point. You really need to look at the dynamics across time if you really want to have an effect on heritability. And so, I'm sorry, Vincent, but this is the new Apsium sorghum model, or Apsium model, and it's a simpler picture, but really this thing just says there's a whole lot of environmental effects. There are some developmental things that determine how many leaves you get when the crop transits from vegetative to reproductive, how it sets up its leaf area, how those things are regulated by these environmental factors. The canopy will then determine the growth rate dependent on the water balance or the light interception, and you'll get some biomass accumulation that will be partitioned to various organs. And so there's a lot of sort of biological sense in this thing that captures a lot of dynamics that are happening as the crop grows and develops and grows. And that's the framework we sort of look towards using. So in Scott's example, the issue here was about looking at early vigour in wheat and using these sort of platform imaging systems to look at tillering. So tiller number, thermal time, and so you've got a high tillering and low tillering line and leaf number and so on. So you can get from these sort of detailed glasshouse or platform measures some indication of key characteristics of canopy development associated with early vigour, in this case to do with tillering. You can also get from the field platform things like, and Bernie showed this the other day, the nitrogen distribution through the profile, so you get some idea of the canopy nitrogen, which will influence light use efficiency. You can then also use these phenocopter arrangements in the field with UAVs to look at flying plots over time, and you get this, gradation, increase in ground cover over time, the leaf area index associated with that, and you can track the cover or the leaf area index of a plot over time. Then you can start to look at, rather than just using the measure that you're getting on that particular instant, you can run that through a model and derive some of these sort of virtual phenotypes or not so visible phenotypic measures, like the stress pattern that plot might have experienced, or its total water use at certain times. So they're much more integrated, and they're much more, they do this dynamic over time. So to do that, you can put these things into the model. Okay, so you've got these measures from your platforms, either in the glasshouse, in the field, or from your UAV, and you can start to get derived phenotypic values that add that integration to what you're trying to do. So the main thing, the outcome of that, you've got a potential to predict new phenotypes like water use pattern by combining those methods with some model that can capture those interactions. The second thing I wanted to just finish on was looking at residual variation and genomic selection, and this is something we're just starting to play with, where we're looking at removing the effects of things we think we know very well, particularly on canopy development, and then considering what's left after that. And this can help in selection in breeding trials, because often a lot of things that are thrown away on the basis of yield are actually high-performing genotypes. They just have the wrong maturity and the wrong touring, which are fairly highly heritable traits that are easy to change. So what we do in here is we've set up in Queensland our own little field phenotyping system and platform that's a sort of version of the Melba one, with a high clearance rig for sorghum and some UAVs, and with some connection to the Japanese groups, we can do this image processing to get head counts out of here, and we can get it pretty well. So we can start counting heads, we know the number of plants, and so we can get tiller number. We've invested a hell of a lot of time getting this data pipeline worked, and so I understand the conversations earlier about things like PhenoSmart, getting these things, the mosaics sorted, everything running, the plot data out, takes a lot of software engineering, and so that system we've now got humming, and so we can start to do things with this. One of the things we're looking at is this residual variation. It's a PhD student looking at sorghum in northeastern Australia. So we've got a number of multi-environment breeding trials from David Jordan's pre-breeding program. And what we do in those, it's a bit like, again, what Francois said, you've got to know something about weather and soil moisture. You've got to have an extra few measures in those plots. We also go in and take a quadrat in the check. So we've got some ground truthing of biomass and leaf area, and that helps us to sort of parameterize the model for the check genotype. You can then, so you sort of template that, but then you can look at flowering time and tiller number on every other entry in the field and generate what would happen if all that genotype had different was its tiller number and its flowering time. And you can then compare that with what you actually observe and look at whether we remove the confounding effects of those two variables, and you can calculate the residuals. And the first crack at that, and hopefully we'll get better experiments this year, this is the observed yield against flowering time, and these are just all the observations from the genotypes in the trial, and this is just sort of the average response to flowering time. So genotypes with a flowering time around 56 to 58 days yielded best there. This is simulating that trial just with the check, but the columns here are the different flowering times. The points of dots here are just adding more tillers on, okay? So within each column, we've got either no tillers or up to 2 tillers, and you can see for a very early genotype, as you add more tillers, you get more yield. But the average for one tiller does this. And so it reflects what we see in this data. But the point is, if you just chose the highest yielding things in here, you're missing the ones that might have been earlier with this number of tillers and yielded up here. And they're the ones that might get thrown away that are really important, or can be really important in the breeding program, so you can really change your selection effectiveness. And I think that was the conversation yesterday about how can this stuff be useful? Well, you can change heritability and you can change your effectiveness of selection. If those two things aren't useful, then you shouldn't be a plant breeder. So the answer around the modelling bit is really just bringing some sort of awareness of biology into this black box and using it in a way that can help decisions that breeders have to make. Thanks.
Okay, thank you, Graeme, and you did a great job of sorting out those 200 slides into the most relevant ones. So we've got time for maybe 1.5 questions. So anybody got a question? Okay. Well, in that case, we're going to step... Oh, there is one. Oh, sorry, Francois, I didn't see. No.
I don't mind.
So, Graeme, was a great talk, as always. So I think you guys are placed in the best possible situation to answer this question, I suppose. Are we at a point to say that what traits across greenhouse and the field are highly correlated and which are not, so that you can actually start to focus on those which are not and try to improve our understanding between greenhouse and field?
Okay, yeah, no, that's a good question. And I think the problem maybe is in the premise of the question in trying to seek a correlation. And it's not really what it's about. So what you're trying to understand in the more controlled environment is what's the variation in a trait that might be relevant in the field. You're probably not going to be even able to measure it in the field, but you'll be able to model the consequences of the variation of that trait. And that's more where we go. We've got some beautiful examples with root systems where we can get nodal root angle of the first flush of nodal roots of sorghum, and that relates beautifully to the water extraction pattern of the mature plant in the field. So, it's not a correlation, it's more like a connection between the relevance of that trait and what it means in the field. And I think Francois gave some beautiful examples of that with the platforms they're using in Montpellier, and Vincent with the canopy development and canopy conductance. So those things, it's once you get into the field, you've got so many interactions going on. You probably may not even see a correlation, but that's not really the point. It's really whether that genetic variation will have an impact in the field environments, and the modelling can help you understand that by unravelling a lot of those interactions. I hope that makes sense.
Okay, thank you, Graeme. Please join me in thanking Graeme for a great talk.